Hey AP students, Dr. Holden. So today is lesson 2, and this is going to be focusing on the organelles that the AP College Board and universities think that more emphasis should be placed on at the college level. And so I've listed those here: the endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosomes, vacuoles, and chloroplasts. And the AP College Board focuses on not just knowing what the structure is and a brief few words about the function, but also how the organelle functions within the cell itself. So that's taking it up a little bit deeper than what we had previously. So with your endoplasmic reticulum, remember that there are ribosomes on there, and the ribosomes, I have not put those up here. Let me add those ribosomes. And ribosomes are either free-floating or they're located on the endoplasmic reticulum. And one of the reasons that we take a look at ribosomes is that every life form on the planet, anything that's considered to be living, has ribosomes. So we're talking about bacteria, we're talking about plants, animals, etc. So the ribosome is important because it has ribosomal RNA, and it is where protein, of course, is synthesized. And they can be, again, free-floating or on the rough endoplasmic reticulum. And the rough part is what we call compartmentalized for the cell. The smooth part, which we really didn't spend any time at all in CP Bio, this is where it is used as a detoxifier, and it detoxifies poisons or medicines by usually adding a hydroxyl group to that. That's one of the reasons why we spent that one lesson reviewing chemistry and some of the importance of what we call the macromolecules, or those large molecules. So the smooth endoplasmic reticulum is critically important in that it adds, when there's a poison or a drug, even aspirin would be considered like this, a hydroxyl unit is added to that to be able to flush it out a little bit easier out of the cell. And that also tells you any place that we consider to have a detoxification in our body, you're going to find more of this cells with the smooth endoplasmic reticulum. So liver is known as a filter organ in our body, and so there's going to be a great deal of that cells with smooth endoplasmic reticulum because they need to detoxify. And as a person is having issues, added medications, or having alcohol or drug, not prescribed drug issues, what happens is that the body adjusts to that. So it keeps manufacturing more and more of these cells with more of the smooth endoplasmic reticulum. And so what happens is that if a person has been on a prescribed medication for 3, 4, 5 years, the body has adjusted to that, and so it has made a great deal more smooth endoplasmic reticulum available, and that means that those even prescribed medications are being removed from the body faster. So I always question when I hear a doctor has had someone on the same medication for 6, 7, 8 years. I'll use one of my relatives. She's been on what I consider to be a questionable medication to begin with. Methotrexate is a chemotherapy drug that is used for a variety of treatments. And she's been on that for over 12 years. And she's starting to have some of the same issues again that she had had prior to being put on the medication. And I said, well, that's because you're on the same dosage. Your dosage level should need to be increased at this point because your body has adjusted to it. So the body really, truly does adjust to medications. It's also the reason why a person who's an alcoholic can sometimes have a great deal of alcohol consumption and not appear as inebriated or as drunk as somebody else, because their body has adjusted to that. So they can actually have a lot more detoxifiers in their body. Whereas a person, for example, like myself, from a cultural background, my genetic heritage being 100% Irish have a tendency to have very limited dehydrogenase. It's an enzyme that removes alcohol through the help of that smooth endoplasmic reticulum. And so if I have just a small sip or two of champagne at a wedding, I am already getting a little bit of a buzz feeling. I already could not be driving a car, and that's just from a few sips because I have virtually no dehydrogenase. So in addition to that, the smooth endoplasmic reticulum being a detoxifier, it is also where lipids, one of those macromolecules, is manufactured. And part of that lipid group are steroids. And so this is where we are finding the steroids of our sex glands manufactured. So estrogen, testosterone, etc. So again, we would find more of this type of cell with larger smooth endoplasmic reticulum involved in the ovaries and the testes. It is also, strangely enough, where calcium is stored. So 3 big functions of that smooth endoplasmic reticulum, which we barely talked about in CP Bio, is that it detoxifies by adding a hydroxyl unit. It is where lipids are manufactured, especially your steroids of the sex hormones, estrogen, testosterone, and it stores calcium. And we spent quite a bit of time on that rough endoplasmic reticulum, but the things that we probably didn't mention is that it is for mechanical support and in intercellular transport. So the endoplasmic reticulum has what is called a lumen, and a lumen is a tube in medicine and anatomy. And so when we look at this, when proteins are manufactured, there's actually a tube that the protein can come out of the membrane of the endoplasmic reticulum. That's especially where the glycoproteins are manufactured here. And those can then be released into the cytosol by having a vesicle placed around it. And oftentimes where they would go to is the Golgi apparatus for sort of a finishing or getting a completion of their job. So remember from that short chemistry overview, we talked about proteins and how they are folded, and some have a simple folding and some have a secondary folding or tertiary folding, and they're held together specifically. And if anything denatures that protein, it breaks the folding. So part of the Golgi apparatus's job is those proteins would come in from the ribosomes and could get repackaged, or the folding could get corrected, and they could be chemically modified for a specific job. So the Golgi apparatus, if you went to college just 15 years ago, would have had a minimal discussion. And just within the last 5 years, they've discovered that the Golgi apparatus really has quite a significant job as far as proteins. Ribosomes manufacture them, but really it would be like here cloth is manufactured and here's where all the clothing is produced. So if you, again, the analogy of cloth, and here's the looms that are making the cloth does nothing for us as far as wearing every day. So this is where the one sheet of cloth could be made into a shirt, whereas another, the exact same cloth could be made into a dress depending on the needs of that cell. And then the mitochondria, we did spend some time. Generally, we do the cellular respiration or the Krebs cycle and photosynthesis towards the end of CP Bio. And of course, we were out on remote. We would have spent a solid week on the cellular respiration and another week on photosynthesis. So we're going to have to spend a bit more time in AP Bio than we ordinarily would, just learning the fundamentals of this once again. So the mitochondria is a double membraned organelle, and the glycolysis for how we break down sugar happens on the outside of that mitochondria, and then it enters in when it's going into the Krebs cycle. And cellular respiration happens in that matrix, or that piece of the membrane in here, the inner complex. And that is where eventually we're going to get ATP manufactured through the inner membrane of the mitochondria. So again, you already know the foundation of this. This is where our ATP is manufactured, or at least most of it. There are a few ATP that are made outside of the mitochondria in glycolysis, but the percentage of them are inside of that mitochondria. And we'll spend a good couple days on just going over cellular respiration and photosynthesis again. The lysosomes, I would have drawn when we first learned those as Pac-Man, and I warned you at the time, I said, don't get that visualization in your head, because they don't look like that. So now in AP Bio, I'm just drawing them as circles. one of the things that the AP College Board warns teachers or professors is that try not to use simplified terms like Pac-Man or the brain center, because oftentimes as they read essays from students, they'll see the same terminology and they won't give credit. So let's leave the Pac-Man analogy in our CP Bio class. And here we're going to say the main functions are they contain hydrolytic enzymes, and that they are for intercellular digestion, recycling, and they also program a cell for death. Apoptosis, it's called. So let's just take the enzymes first. So we know that lysosomes contain what they consider to be digestive enzymes. And so there's at least 50 of them. It's assumed that there are somewhere between 52 and 55 of them. There could be more. By the time you end up into grad school, you could end up knowing that there are 60 of them. That's just how quickly research goes. And so these digestive enzymes are responsible for specific breaking down of items within the body. And if a person is born with missing one of those, a gene that will manufacture one of those enzymes, there'll be a genetic disorder. And an example of that is Tay-Sachs. So in a baby born with Tay-Sachs, they would have received the gene from both mom and dad. And the baby appears perfectly normal in development all the way through, usually until about month 5 or 6. So usually the baby right around 6 months of age can start sitting up by themselves. They are starting to roll over. And then very quickly, the baby seems to degrade, where 1 week they're sitting up and the next week they can't sit up by themselves. And parents have a tendency to prop up and here, practice this, and the baby just flops over. And genetic testing would be showing that they have Tay-Sachs. And that is generally from people of Jewish descent, where culturally, decades ago or hundreds of years ago, even if you look at Russia, Germany, Poland, in Russia, for example, the Jewish population were confined to villages, and so the ability for people to marry was limited, and so there were genetic abnormalities began. It's just similar, for example, in my own culture, being again purely 100% Irish. I told students in CP Bio that a very strange sickle cell anemia, which is considered to be an African American disorder, has been found over 20 years ago in a population of Irish in Chicago, where they had been brought over from Ireland by a group of very wealthy landowners in Chicago, and the landowners built them schools and churches, etc. And the population of the Irish were very happy and did not leave, and they ended up marrying other people that had come over with them. And so again, the gene population was very limited and caused a genetic anomaly. So lysosomes have those digestive enzymes, which are important as far as normal brain and body functioning. In the case of Tay-Sachs, what happens is a lipid builds up in the brain, and they don't have the enzyme to break that down. In addition to the digestive enzymes, it is also known as a intercellular digestion. So it would be breaking up toxins or other materials within the cell through a system called phagocytosis. And that's where I had that analogy of where the lysosome is going in and grabbing those pieces, things that are no longer needed or waste products, etc. And if the waste product is too large, they assist it through endocytosis of getting it out of the cell. So digestive enzymes, and they act as intercellular digestion and recycling. And the third thing is that programmed cell death, apoptosis. So lysosomes can help trigger when a cell should no longer exist. If the cell recognizes that it's abnormal, if it's aging, if something has gone wrong and mitosis did not stop, then apoptosis should take place. And we'll be talking a little bit about what the whole program is of cell death. And then we have a variety of vacuoles, and they are for storage. They could have macromolecules temporarily stored in there, and then they would be released as needed. They can store waste products. In plants, we have that large one general central vacuole, and that would be only 100% for water storage and for what is called turgor pressure, or keeping that plant nice and firm. So again, vacuum